Yes, that's exactly what we use is like this, assays that directly measure the reactive oxygen species present.
Hmm.
So that means it has to be produced by the cells. Now, we also looked at markers of oxidants, right? So we found that NOx4, which is a master regulator of oxidant production in many cell types, that's its only known function is to produce oxidants. NOx4 levels were very, very low in these psilocin treated cells. And NRF2, which is a master regulator of hundreds of antioxidant gene responses, NRF2 levels were extremely, so you had, the net effect is you have much greater antioxidant responses and much lower oxidant production. And that net effect is reduced oxidative stress.
Got it. So markers change such that in the presence of psilocin, there were fewer oxidants and more antioxidant production. So basically, the cell can tolerate more oxidative. It can tolerate more oxidative stress, or it can protect itself from that stress for longer.
Right, exactly. Yeah.
Interesting. So what about like telomere length or anything like that?
Yeah. Yeah. So telomere length was preserved. So whereas again, we normally see that classic hallmark of aging that you see telomere attrition. So telomere length is shortening over time. So in cells that did not, that received vehicle treatment, we compare these back to the same cells as young cells. We can see that telomere length was much shorter with normal aging, and that shortening of the telomeres did not seem to happen to the same extent in psilocin treated cells. So there was so that telomere length was preserved when you treat with psilocin.
Mm-hmm. So you looked at a number of things: reactive oxygen species production, the expression of regulators of oxidants and antioxidants, telomere length, a lot of the major things one would look for.
Senescence.
Yeah,
Yeah.
yeah. And so basically across the board,
Yeah.
psilocin made these cells less senescent by increasing antioxidant stuff and decreasing oxidant stuff.
Sure, yeah. And then, we see these other and so now the question is like how are these things working? Because we don't think, for example, we have some unpublished data showing it's not acting directly on NOx4, but the downstream impact is on NOx4. So
Mm-hmm.
psilocin doesn't, we don't think it's binding to NOx4 and doing something. The downstream effect is that it's impacting these pathways, right? So we don't know the order of what's happening first
Right,
and
right.
how these things are
Yeah.
interplaying.
The end effect is this
Right,
anti-aging effect. You know that
right,
psilocin is getting in there somehow, but it's a bit of a black box in between.
right. Like for example, is it the fact that you have less oxidative stress? Is that why the telomeres length is preserved? Or is it the fact that you have more, or is it the other way around? Do you have more protection of your telomeres?
Right, right.
And that's leading to more efficient DNA repair, and that's what's causing the net effect of less oxidative stress. How those things play into each other, we're still kind of teasing out. So we
Yeah,
don't,
yeah.
but the net effect is that you have that the cells will age with psilocin treatment, but just slower.
Mhm.
So you still have the cells will still become senescent and they will reach the peak of their replicative senescent lifespan, but just that process is delayed when you treat with psilocin.
Right. And so a natural question at this point is if it's having this effect in a dish, would it have this effect in a whole animal? And so what did you do there?
Right. So yeah, we were completely excited about these findings in cells, and so of course the next step is, well, we need to test this in animals.